Hey there. So we are going to fill in our notes from packet page 88, Introduction to Genetics Notes. I have already cut out the notes and glued them in to my notebook. So let's go ahead and get started. So when we're talking about genetics, right, it's a little bit of a review talking about DNA, talking about genes, right? So remember that genes are directions in the DNA for making proteins, right? We talked about protein synthesis, right? Grabbing a small chunk of the DNA and reading it and turning it into a protein. Well, that's what a gene is. It is that one recipe for making a protein. Okay, so the gene is the recipe, and of course, DNA is the recipe book, which has all the recipes. Most of our genes come in sets of two. All right, we get one set from mom and the other one from dad, and then these two copies of the genes, right, like what we did in our colorful karyotype, right? You got one color from mom and one color from dad, but these two copies are called alleles. 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 An allele is a version of a gene, right? So if we think about the different traits, that affect how we look, right? We can think about hair color, skin color, height, hitchhiker's thumb. Is your earlobe attached or unattached? Can you roll your tongue? Eye color, right? Anything that makes you, right, is a gene. And there are different versions of those genes, right? Some of us have blue eyes, some of us have brown eyes, right? So on and so forth. So the genes that an organism has, so, right, all of the genes that I have is called my genotype, right? And I got one version from mom and I got one version from dad. So an example, right, of my genotype would be the letters that I use, right? So big T, little t, right? So maybe mom gave me an allele that would make me tall. Dad gave me an allele that would kind of make me a little bit shorter, right? But so it's the same gene, just a different version. So I got two alleles or two versions of the same gene. In this case, maybe we're talking about height. What that is physically going to look like is called my phenotype, right? What I'm actually going to be. So in this example, right, I would be tall, right? I have a big T. So maybe tall means I'm above, 5'7". I think that's the average human height. But height is a little bit more complicated than that, but that works for this example here. So when traits actually show up, right, there are two versions, right? Sometimes we can say that a trait is dominant, meaning that it always shows up, right? I only need one copy of that allele for that trait to show up. And we represent these traits with an uppercase letter, right? So capital T, capital A, whatever. It's a capital or an uppercase letter, okay? The opposite of a dominant trait is a recessive trait, when you have to have two copies of that allele for it to show up or be expressed, right? And this is going to be represented by a lowercase letter. So in this example, in order for me to be tall, right, I only needed that one capital T. But in order for me to be short, right, I would need two copies because it's recessive. Okay, so I would need two of the lowercase letter T's. But to be tall, because tall is dominant over short, I only needed one copy of that uppercase or that capital letter. Okay? When we're actually looking at the letters themselves, right, the alleles, there are two different ways we can represent the two alleles. If the organism has two of the same alleles, we say that it is homozygous, right? Homozygous, the organism has two of the same. So, right, I could say big T, or little t, right? That's homozygous. If the organism has two different alleles, we say that it is heterozygous, right? Big T, little t. Okay, so homozygous, both the alleles are the same. Heterozygous, the two alleles are different, so uppercase and lowercase. So when determining the chance that you will inherit a gene, right? That's your probability. So like in our karyotype, right? We talked about independent assortment, and we flip the coin to determine which chromosome we would get from mom and which chromosome we would get from dad. And in order to determine the actual probability of inheriting a gene, we use a Punnett Square. Right, this is a device that geneticists can use to determine, right, what is the actual chance of an offspring inheriting a gene from their parents. So, for example, right, let's say, right, mom, right, she's tall, but she's heterozygous tall. And then let's say dad over here, right, he's tall, but he's homozygous tall. So I can use this Punnett square, right, which is basically the FOIL method from math, to determine, right, what is the chance that the child from these two parents would be tall, or what is the chance that a child from these two parents would be short. So Punnett square is just a tool we use to determine the probability of the offspring actually inheriting a trait from their parents. So that's it for these notes on our introduction to genetics. Make sure that you have cut out packet page 88 and we've glued it into our notebook. If you have any questions about these notes, just make sure to let me know. Okay, thanks.